that's here, and then I can give him the folder.
Yeah.
But, oh yeah. Yeah, it's all good. I said, you saw I sent an email out to everybody and said, oh, we're gonna start a little late.
Yeah.
They'll just give you a little.
Yeah. Dan still was like, well, he emailed me again and was like, can you start without me anyways?
No.
But we can't.
Yeah.
Okay.
All of the committee has to be here.
Okay.
That's why I was like, how late are you gonna be? Because if he's gonna be like really late, then we would have to find somebody else.
Yeah. Okay. Hey.
Hey. So that's why I was like.
Yeah, just 10 minutes. Hopefully.
Well,
Yeah.
but you were. I said.
Hopefully it doesn't slide.
Oh, actually, Erin and I ran into each other. So we actually wrote down together.
Oh, that big grin.
And you lived downtown. I actually just done this.
I know. He's like, I'm done. Now. You had a big grin before, but now it's even bigger.
That's good.
Okay, well, I'll just kind of hang out. Yeah. You're all good. You're
Yeah.
pretty relaxed.
Yeah, on the outside.
On the outside.
Yeah.
My
Oh, trick?
trick.
Oh, do I have a trick?
I do.
Oh, what's your trick?
I just like.
Primarily, actually.
But like,
Slowly.
your box.
Rather than,
For 4 pounds.
Oh,
Excel for 4 pounds.
okay.
Hold
I
it
think
forward.
I lost in 2016.
So
It
this-
might have been.
Yeah, it works like
Okay.
a charm.
I think I've heard of something similar. Or like,
Yeah,
was
there's like 478,
5 years
yeah, but like 4444,
Oh,
it
yeah.
works like a charm. I was at the DMV and I was having like a total stress face. And
Wow.
I started breathing and nobody knew, like that's the thing, nobody knows that you're doing it. And worst case, you just make your exhale longer than your inhale.
Yeah. I'll do that. I think once 2:30 hits, also my nerves will start to kick in more.
Yeah, you didn't have any coffee.
No.
And then you,
Yeah.
actually need to approve.
Um,
It's like in some ways life slows down and other ways it speeds up.
Yeah.
Did you go out and celebrate?
Okay.
Did it?
Well, yeah, you did.
Right, or even just.
Yeah, I know.
I know,
right?
Yes, exactly.
Yeah, Katie and I like, we divided the comment today. She took the morning ones and I took the afternoon.
There were 4 today. By the end of it, you're just like
9, 10.
Yeah.
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 Remember.
It's my afternoon thing,
Yeah.
All good.
So you're doing, bio
It's a little bit more. It's a lot cooler in here than it is outside.
What's that transition? The first year was kind of difficult. Anyway, so I knew I wanted to come up. Didn't want to go to the southeast. I was hoping. Too many. A lot of cars all the time, so it took a while, but I thought do something. Yeah, it's just something beautiful. One of my friends was in my grad school with families and do something.
You ready? Yeah.
And it's a really pretty good university. Yes, it's a really good university. There's lots going on there, music, theater, art. I cycle, so they have cycling. And I travel. So, yeah, the journey is good. It's really
Yeah,
good.
yeah.
So who's late then? Who's late this week?
One of the committee members.
Yes.
Your dad, oh yeah, your dad does have a Facebook.
Painting exhibition, yeah. Yeah. Body, they have to create body.
Does anybody have gum?
Fine, sure.
No, I don't. Sorry. I thought they did. Hey. Have
We're
mouthwash.
starting a little late.
Yeah.
Hello. Thanks for coming. You're the earliest.
Well, I'm just the chart, so I forgot to drive. I'm supposed to be bringing the.
I have
Okay.
Oh, you're out.
I have the folder. I'm Deborah. Hi. So this is the folder. I think, did I see you last week in the defense?
No.
No? Oh, okay. So this is the folder. It's got,
Needs everyone.
needs your signature,
Just
actually,
mine.
just yours, but all the appropriate boxes checked.
Okay.
And then, if you could either send it back with Jennifer or Kosu or Christina.
Hey.
Yeah, you can't bring it.
One time I see.
Hey.
There's actual ballots in there, yeah. Yeah, you have everything. Yeah, so everything is in there.
I'll send it back to
Send it back with.
Yeah.
Yeah.
Are you gonna be here this summer?
I will.
Yeah. Are you guys gonna be?
No, I have a writing
Yeah, he's
quarter.
gonna be.
I have a writing quarter. We should go lunch
Um,
sometime.
Oh, yeah.
Saying that we're gonna start a little bit
And
later.
Ben has
because we're
a
gonna
dental
be here
school.
until like 2:40.
So far so good. Yeah, almost done with the
Something else.
First year.
third quarter. Yeah. Almost done with the first year. Yeah.
Right.
Mhm.
What time?
It's 3 years or 4 years?
4 years.
4 years.
Yeah.
Most schools are 4 years.
So it's just gonna like
The other thing-
shorten it, yeah.
Yeah, just the other thing.
Shorten it.
Anyway.
Okay, yeah, sorry.
Yeah. No, that's fine. You're
Yeah.
like
Oh, that's
definitely.
You got
Okay,
it.
definitely catch up later.
40. In the building? He's coming from. It'll.
Yeah.
Same seat.
Yeah, for like,
He has to leave like on time. So it's gonna. Cross. Yeah. Hi. Nice to meet you.
No, I need something else.
Oh, you have to leave something else.
That's a long time ago.
All right.
Yeah, it is a long time ago.
Here, I have,
You would like to. Oh my god, no, it's not.
I wouldn't go. I would just refuse to go.
I don't think I'll be.
You're just being denial.
It's like, no, it's
That's fair.
So you're from New York.
Yeah.
Okay. Okay, nice.
I know the place was all depends on the same situation. Tell me why.
Is that the only reason you're excited to go? Because it's how
Yeah.
I'm just wondering.
Are you a grad student here now? Oh, nice. And what department? Oh, wow. So you were smart enough to learn science.
Well, yeah, I don't know. Econ is also, I'm not going doing it over again. I probably would not have gone into econ.
You don't think so? Would you consider it?
I would. I find economics culture to be very toxic though.
Yeah.
So I'm not sure if I would. I would definitely think twice about whether I wanted to. So,
Huh?
Here.
Of course I'm here. David, how are you?
Hello, good to see you. Long no see.
I didn't Very excited to go. No pressure. This better be good.
I have handouts.
Oh, great.
Still, yeah.
At least you know I don't.
So you left me back.
I love it.
Hi, I'm great. How are you? Good to see you too. Wonderful. How are things on the exciting.
No, that's
Yeah, it's good.
Oh, good. Yeah. Oh, this is you were in
Yeah.
Yeah.
Well, to me. Did you guys end up hiring?
So we did
Yeah.
we're gonna just have a skate house. We have like 10 people now, but we have a bunch of consultants, but we're looking for looking into space. But you know, my company's
One other.
No, yeah.
Notice was on fire.
Yes.
So they merged with. So
So now we have now we have a big
okay, from the aisle to. Are you already? You're not. Okay.
Based on
Oh, is that right?
That's exciting. Yeah, and then we have a new technical resale street.
Yeah, we stopped at
Different than.
Well, now it's on steroids.
Like a million. For invites. For fun.
Yeah, we should have.
Yeah, let's do it.
Are you
Now if you need.
Oh, yeah?
Oh, yeah. John doesn't.
Oh,
Yeah.
well, yeah, let's do
As
that.
long as it's not competing. Yeah.
But maybe when they come to it.
Maybe we could.
Yeah, exactly.
Oh, geez.
I just
Theoretically, out of point on the-
I'll make sure to tell you.
Oh, you did that?
You did send me trying to outstate me like cigarette. You haven't had my
And so. Like in the whole.
I know.
No.
Oh, it's in your place. Oh, I didn't know that. I was wondering why you was no one else was, and it's like, okay, yeah, that makes sense.
We got like 5 more minutes. Okay, just waiting on one more member of the committee, so keep talking.
But no one will. Every time I cook for it, but never eat it. But also, especially like, I made that mistake. Oh, yeah. I made my gifts and I put out syrup and it went from like one person putting a dollar box to like one person putting less. That's when they got the
So maybe they do. You're right, because I'm gonna use
Just one. Yes. Like, he wore that yesterday. I was like, go right in the field. And I also wore these shoes.
We always do.
I figured he was gonna wear his
Oh, so you guys.
Yeah.
At the same
Yeah. Oh, for the. There was something. No, it's you can put your face on.
We're still waiting.
You could do this on the introduction.
Just give
I
me the
could,
three
but
of
then
them.
if I finish, and do we want to continue with that?
Then someone can do some singing without.
Yeah.
Maybe more awkward.
I enjoy it. I miss.
I'm sure you're doing
Not too bad. Oh, yeah.
Yeah.
So.
I thought we were.
I'm sorry. I'm sorry. I'm sorry. I'm very sorry. My apologies.
Yeah.
About the machine. Telephone.
Yes, yes. I really like that.
I see it backwards. Sending a letter across fiber optic cable to Australia.
Yeah.
Yes, we done cool
Wait,
Yeah, over messenger.
How good are those? When do you guys start? Like, actually.
We're doing that already. You just talk to those.
Yeah, you're right on.
Well, it was.
He is very confident. One day I was making some class, he said he was offended that I funded it.
He's right, but you're right. He's trying to be really
I realized that.
These are all
Going on. And this
actually no.
Football.
I know he's gonna. Flexibility is.
Yeah. So we can start.
All right. Good afternoon. I would like to welcome you all to Aaron Craven's Thesis Defense. I'll give a brief introduction. Aaron received his bachelor's degree from Rochester University in applied mathematics. As an undergraduate, Aaron conducted research across laboratories in physics, chemistry, and structural biology, highlighting his truly interdisciplinary training and interests. His quantitative background spanning biology, chemistry, and physics made Aaron an excellent fit for Stanford's bioengineering program, and he joined our department in fall 2014. So when joining my lab, Aaron expressed early on his interest in evolution, and the engineering of evolution is a key strategy in the bioengineer's toolkit to enhance and create new biological activities. Taking inspiration from nature's approach in the immune system, Aaron has developed and led his thesis project to reconstitute B-cell somatic hypermutation in yeast. As challenge after challenge came up in Aaron's project, he approached troubleshooting his system and what I would say is a very typical Aaron fashion, taking a step back, calmly and deeply thinking through the issue, and coming up with a creative solution to address the problem, all with a very positive and optimistic manner, while then wasting no time moving on to the next challenge. The result is an in vivo mutagenesis system with substantially higher on-target mutation rates than recent state-of-the-art systems have described. Now on the side, Aaron has contributed to 3 separate metabolic engineering projects in the laboratory, enabling yeast-based biosynthesis of diverse plant natural products, including the purine alkaloids, noscapinoids, and developing a novel biosynthetic pathway to papaverin and related 1-benzoisoquinoline alkaloids. And he actually won't be sharing any of this work with you today. His contributions to these projects demonstrate his versatility, really incredible ability to multitask, and the high regard that his colleagues hold for him as a collaborator and scientific contributor. Now, speaking of multitasking, Aaron has spent over 2 quarters interning at local biotech companies, Google X and Andea, and during these internships, he's basically continued to moonlight in the lab at Stanford, pushing his thesis project to completion. His work ethic, focus, and efficiency are truly amazing, and continuing that general theme, Aaron will be spending the summer balancing wrapping up his paper and thesis, participating in Stanford's Ignite program, continuing working as a consultant at Andea while making final decisions for his next stage activities. So Aaron.
Great. Well, thank you, Christina, for the really kind introduction. Hopefully my presentation lives up to all those words. So, like Christina mentioned, my PhD has been focused on developing tools for engineering biology. And in particular, I've been focused on developing tools that harness evolution. So, nature evolution is the changing characteristics of the species over generations. And evolution is responsible for all the biological complexity and diversity around us, hinting that it might be a very good tool for engineering biology. A simple example of evolution in action might be the evolution of something like mouse coat color, where genetic variation in a population leads to slight differences in coat color, which if perhaps these mice live in a dark environment, mice with darker coats are then more likely to survive and be selected, and if this is repeated many times over generations, new traits can emerge as a result of this cycle of variation and selection. And it turns out humans have been harnessing evolution for thousands of years by directing it in the form of selective breeding. For example, corn and all of our staple crops that we use today have ancestral equivalents which were evolved over periods of hundreds of thousands of years long ago. And this is also true for many of the animals that we use throughout society. But notably, these efforts took hundreds and thousands of years to achieve. More recently, with the advent of recombinant DNA technology in the 1970s and 80s, we've developed methods to speed up the process of evolution in the form of directed evolution and being able to artificially generate diversity. And this has resulted in the advent of evolved biologic drugs like Humira, which is used to treat arthritis, and even enzymes which have been evolved for cleaning products and for use in green manufacturing. You may not know, but if you have laundry detergent at home, there's likely enzymes that have been engineered in that laundry detergent to work in cold water and remove stains such as protein stains and food stains from your clothing. And so the technique responsible for these latest advances and a rapid speed up in our ability to engineer biology is a technique called directed evolution. And directed evolution is just the iterative application of mutation selection. It typically consists of a cycle like this, where one has a gene of interest representing perhaps enzyme or antibody. This gene is artificially diversified outside of a cell in a process called ex vivo mutagenesis. Following artificial diversification, the DNA library is transformed back into cells to express protein or whatever trait of interest, followed by selection or screening for the desired property, whether that is an improvement in some sort of enzyme activity or a change in binding. Following screening of cells and selection of variants that have improved activity, DNA is re-isolated and this cycle is repeated many times, slowly walking in the direction of the desired level of purpleness in this case, or enzyme activity, or binding, or whatever property is desired. And so this is the technique that is responsible for all of the recent advances in engineering biology, and is used universally in industry and academics to engineer biology and evolve things like enzymes and antibodies. And most recently, the originators of this technique, Francis Arnold and others, won the 2018 Nobel Prize for their work in developing this field. And yet, despite the massive success of this method, there are still many limitations. In particular, the need to isolate DNA, artificially diversify it, and transform it back into cells. These are very labor-intensive tasks. There's practical on how quickly these steps can be carried out and how much diversity can be generated. And for these reasons, recently there's been focus in the field on trying to develop methods for in vivo mutagenesis, which can allow us to replace the entire right half of that cycle, and rather than removing DNA from a cell and artificially diversifying it, by simply growing cells, we can create diversity within our trait of interest. The benefits of this include it allows us to construct systems that are amenable to continuous evolution. Obviously, as you can see, it vastly speeds up the cycle of evolution, shorter time periods and zero labor, because the cells are doing the work. It's amenable to automation, and finally, one can potentially generate unlimited diversity because there's no bottleneck of transforming DNA into cells and getting it back out. So with this in mind, the challenge of constructing a system for targeted in vivo mutagenesis consists of the following. Within a cell and within the genome, we have a trait of interest encoded perhaps in chromosomally, as shown here, or episomally on a plasmid. And that trait represents just a small fraction of the entire DNA in the cell, perhaps 1/10,000. And the characteristics we would like to have in a system for in vivo mutagenesis are, of course, targeting. We want to target mutations to our trait of interest and avoid mutating the genome. Of course, mutations in the host genome are potentially deleterious to the host and can be problematic in the downstream selection steps. In addition to targeting, we would like to have some degree of control over the mutation rate, such that we can tune the degree of diversity that is being generated during the in vivo mutagenesis process. And finally, because DNA consists of A, T, C, G bases, we would like to have diversity at all four of those positions in order to generate sufficiently diverse sequences. And so, others in the field have constructed systems recently that embody some of these characteristics for a system of targeted in vivo mutagenesis. A very common strategy that's been tried recently is using the Cas9 binding protein, DNA binding protein, to guide mutagenic enzymes, shown here in red, to a particular region of the genome. By guiding these mutagenic enzymes to a particular location, mutation rate can be increased within around a 40 base pair window adjacent to the Cas9 binding site. This plot is just showing the mutation rate on the y-axis with the genomic position on the x-axis. Where the guide RNA is binding, you can see kind of an explosion in the mutation rate at that particular site. But the drawbacks of systems like this is that, like I mentioned, the mutation window is rather small per binding event. And in addition, to tile an entire gene, that requires the guide RNAs being expressed across the entire gene. So a small gene like GFP, or something larger like a 1 kb gene, could require upwards of 25 guide RNAs to be expressed simultaneously. And each guide RNA has its own binding site, which inhibits or prohibits mutation at that site. And so looking beyond strategies like this, it turns out nature has already solved the challenge of immunogenesis for us. This occurs in the form of a process called somatic hypermutation. And somatic hypermutation is responsible for our adaptive immune system's ability to evolve antibodies on the fly to new pathogenic threats. And what somatic hypermutation does is it turns up the mutation rate at a very precise location in B cell genomes so that antibodies can be evolved in response to new pathogens. In addition, the process of somatic hypermutation has all the characteristics that I just described for an immunogenesis system. It's targeted, it's very specific. The mutation rate within the immunoglobulin locus where mutations are targeted is around 10 to the minus 3. And for context, the global genome mutation rate in B cells is typically around 10 to the minus 8. The mutation rate has some degree of control. Finally, there's great mutation diversity. And so the way somatic hypermutation works is an enzyme called AID, or activation-induced deaminase, deaminates cytosine residues to uracil. And there's a variety of mechanisms that target AID to the Ig loci in B cells. Following accumulation of uracils within the Ig loci, a variety of error-prone repair mechanisms kick in that then diversify adjacent bases in addition to the cytosines that were originally deaminated. Typically, these uracil mutations in a normal cell would be repaired by error-free repair mechanisms, but B cells have evolved methods to alter the error-free repair. And I'll go into those a little bit later. And so, hopefully the title of my talk now makes sense. What the goal of my thesis is to reconstitute this process of B-cell somatic hypermutation in yeast and apply it to directed evolution. And the reason we chose yeast as an organism to reconstitute this process in is because yeast is widely used in industry and academic settings as a model host and as an engineering host. So the rest of my talk will be structured as follows. First two sections will be on developing tools for targeted mutation using mutagenic deaminase enzyme, followed by efforts to enhance mutation diversity with DNA repair modification strategies. And then finally, we take all the engineering that we do to create the system for in vivo mutagenesis and speed up the cycle of evolution and demonstrate that the system is able to work. So onto the first section of this talk. Our strategy from the onset was this. We took the enzyme AID, here actually an homolog CDA1 from C. lamprey, which is a hyperactive AID variant, and we fused it to the T7 polymerase with the idea that by putting a T7 promoter in a particular region we're targeting, it would transcribe along the gene and deposit uracil mutations along our target site. We then realized that these mutations could be processed in several ways. There could be error-free repair, error-prone repair, and of course replication, which would result in CDT mutations. Because we're trying to measure a mutagenic process, we needed a way to measure mutation rate in yeast. And so the assay I'll be discussing throughout this talk is a very classic technique called fluctuation analysis, where one has a model gene they're targeting. In this case, URO3 will be our target model. And if the mutation rate is high enough, there will sometimes be loss of function mutations in URO3 that break the gene. If there's no mutations, the gene is still functional. And when we plate these cells onto media in the presence of a drug, genes that have a broken copy, or cells that have a broken copy of URO3 will be able to grow, and if there's no mutations in the gene, no colonies will grow on those plates. And if one does this to many cultures in parallel, the numbers of mutants arising on these plates follow certain statistical patterns that can be modeled using a certain algorithm and give us a quantitative mutation rate estimate. And as we might expect, if there's more colonies forming on plates, that indicates a higher mutation rate, and we get a higher quantitative estimate. So jumping back into the system I just described, we initially tested 4 different arrays of T7 CDA1 targeting system. We expressed CDA1 by itself. We expressed it in the presence of the T7 promoter to target. We expressed the fusion protein, and we expressed the fusion protein with T7 to target, the T7 promoter to target. And when we looked at the mutations forming on this fluctuation assay, you can see only in the far right column do we have this really significant increase in the number of colonies forming, indicating that we're indeed getting specific targeting in that 4th case. When we quantify this using the algorithm I described, you can see that, again, the first three cases are pretty much indistinguishable, representing the nonspecific activity of CD1 in the genome deaminating at random. In the fourth case, you can see, because we have the T7 promoter present targeting the polymerase, we get around a 60-fold increase in the mutation rate. And this 60-fold increase is something I'll refer to later as the on to off target ratio. And we want that to be as high as possible, because the higher the on to off target ratio, the lower the number of off target mutations we are accumulating. Another way to look at this is if we had no targeting at all, for every single mutation in a target gene, we'd have 10,000 off target mutations. With our 60x ratio, for every one on target, we're getting 170 off targets. B cells go way beyond this. For every single on target and somatic hybrid mutation, there's 0.1 off targets. So we made some progress, but we're dramatically doing worse than what SHM is able to do. And so we want to optimize the on to off target ratio. We tried several different strategies, including altering the linker length, just adding DNA binding domains to assist in T7 recruitment, and also splitting T7 and CD1 and associating them via a modular peptide association strategy. It's this third strategy that gave us some improvement. So what this represents is we took T7, no longer have the fusion to CD1, and we put this SH3 protein domain, which will bind to SHL of the cell. We then fuse SHL to CD1 so that these two can reversibly associate. And when we express them together, you can see we have about a 250-fold targeting rate. But we weren't done. The reason we had this big improvement, I noticed, was because the off-target rate has decreased so much compared to expressing of CD1 alone. And so we thought, perhaps by tuning the amount of the expression of T7 CD1, we could increase the on-off target ratio even further. And so this led us to a fourth strategy where we simply put CD1 under control of an inducible promoter and integrate it into the yeast genome, allowing us to really tune down the amount of CD1 expression, hopefully reduce that off-target rate of mutation. When we did this and look at a range of induction levels for that promoter, you can see that one level of induction in particular gives us around an 1800-fold on to off targeting ratio, which we were really happy with. On the far left, to the far right, at maximally and non-induced, you can see we have about 100-fold control over the mutation rate with the penalty of having an increased amount of off-target mutation. And again, just to put this in the context of what SHM achieves, for every single on-target in our system, we now have 5 off-targets. And SHM is getting a 1 for 0.1. So we're not quite there, but we're doing much better, and we were happy with this. And now switched from trying to optimize this to looking at what exactly was going on at the genomic level. And so to look at what's happening at the genomic level, I turned to a different assay, very deep sequencing. And what these plots are going to be showing is we have an original base, A, T, C, or G, and then we can look at specific mutations to the mutated base. And we can zoom in on a particular type of mutation that's occurring. In this case, we're zooming in on the C to G mutations. And you can see that for our control and sample, we have this overlap of signal. And each dot represents a single base where we have an estimate of the mutation rate. And there will be some noise built into this assay because the NGS method that we use has an inherent error rate. And so taking this approach and looking at the data, looking at the system we had built, we can look at what sorts of mutations are occurring. And you can see there's only one location where the blue curve deviates from the yellow, and that's in the C to T mutation segment. And when we zoom in on that, we get this really pretty curve of the mutation rate at a single base resolution, where the yellow curve represents no T7 promoter present, and it just keeps going along. There's no change in mutation rate on the y-axis. Whereas when we have a T7 promoter, instantly following that, we have this jump in the mutation rate representing the activity of the polymerase binding and then transcribing. And notably, the mutation rate doesn't change because T7 is processive, so it will just keep going until we put a terminator and it falls off. For context, this data comes from an equivalent set of samples that's highlighted here in the fluctuation analysis. The only thing I want to point out here is that in the fluctuation assay, we're able to get a lower mutation rate than we see in the NGS assay, and that's just an inherent limitation of the NGS that we carry out. So we can zoom further and just look at the site of T7 initiation. And you can see that within the promoter, there's not really a variation between blue and yellow dots, indicating there's no mutations being generated there. As soon as we hit the transcription start site, you can see the blue dots pop up above the yellow for the most part, indicating that immediately following transcription start, CDA1 activity is resulting in accumulation of mutations. But all of this aside, we've been able to look very carefully at what sorts of properties our T7 system has, but we were really curious about what other mutations might be occurring beyond just C to T transitions. And looking at this plot, there's nothing apparent that sticks out. Another way to look at it is just breaking things down by single base substitutions. And the only thing that deviates, again, is the C to T mutations. A to N, T to N, or C to R, G to N don't really have much deviation, really hammering home that our diversity is quite limited to one type of substitution. And so this brings me to the second component of my thesis, where we wanted to enhance mutation diversity to correct this problem. In the first part, we returned to the mechanisms that B cells use and took a closer look at what's happening in the error-prone repair processing segment of somatic hypermutation. What B cells do following accumulation of uracil within the IG loci is the uracils are processed following traditional error-free repair mechanisms such as mismatch repair and base excision repair, but rather than using an error-free polymerase, B cells will use an error-prone polymerase to backfill during the error repair process, and this results in mutations at other locations around the uracils. And decades of research in B cells and animal models has identified a list of genes in B cells that, when knocked out, alter the AT mutation rate in B cells. And so this gave us a list that we know these are involved, but we don't know how they're targeted, and we don't know exactly what's going on, but we know that they're involved in accumulation of AT mutation in B cells. Unfortunately, because DNA repair is a very ancient process, the yeast have identically named homologs, which is nice because you can just find the name and not look for these weird uniprot swaps. And so we were able to just identify a list of these homologs and say maybe if we target those, that will alter the mutation outcomes that we're achieving in the yeast. And so the strategy we chose to carry out this targeting of these repair proteins was the SHL-SH3 system that I described earlier when we were optimizing the on and off target mutation rate. So shown here in pink, and pink is the SHL-SH3 domains. We've now fused two SH3 domains to the CDA1, and by putting an SHL tag on any protein of interest, we can associate it to our uracil track along the target gene. And the idea is that we can now push towards mismatch repair or error-prone forms of repair, rather than just replication of mutations from C to T. So we selected around 20 genes from the list of yeast homologs, and we just edited in SHL tags into these genes in the yeast genome. And of the 20 that we analyzed via NGS, only one really stood out significantly, and that was the MSH6 fusion, which MSH6 is involved in mismatch repair. You can see in the bottom right corner, the G to A mutation rate, the blue curve is deviating from the yellow, which we hadn't seen before. And when we zoom in on that, you can see in the absence of any targeting at all, the yellow curve, there's no change in the mutation rate. With no SHL tag, but with targeting in gray, there's a little bump, but not really anything changing. When we have the MSH6 with SHL, we get a big bump in the mutation rate in the G to A substitutions. The mechanism for why we think this is happening is accumulation of uracils on the forward strand of the target gene is we know that's happening. But by targeting MSH6, we think we're increasing the amount of mismatch repair that's occurring. And when mismatch repair occurs, it will chew back the uracil containing strand, exposing the template strand as a single-stranded DNA that can now be deaminated by the deaminase enzyme. And so the deaminase is still responsible for these mutations. It's just a result of an increase in mismatch repair that's opening up another strand, the opposite strand, for deamination. When we look at other mismatch repair associated genes, all of them have the same trend. MSH6 had the most significant signal. And so this kind of gives us confidence that this is the mechanism. But as engineers, kind kind of had to keep moving because the goal was to increase the AT mutagenesis. So we didn't really take a closer look at the mechanistic details. We moved on to combinatorial targeting, thinking that if we're able to get an alteration in mutation rate using just MSH6, maybe combining a few different targeting SHL tags together will give us additional signal. And so we're going to move to a slightly different assay. Now, instead of using NGS to look at these combinatorial strains, we're returning to fluctuation analysis. And rather than looking at breakage of the URF3 gene, we can look at fixing of it. So we installed a TAA stop codon in the URF3 model gene that we've been using this whole time. And if a mutation in A or T occurs in the stop codon, we can have restoration of the URF3 gene, and now we're able to see colonies on plates. So just kind of reverse of the assay I was describing before. And so when we look at some of the combinatorial strains, there was one that really stood out again. Here, YHM2 is just the base T7 strain expressing CDA1 T7. With the MSH6 tag, you can see there's a little bump, but it's not terribly significant. And just as a control, we're putting random peptides on these as well, so that we're checking that it's not just addition of a SHL peptide that's disrupting MSH6 function somehow. But when we combine MSH6 with APN2, we got this really big jump in AT mutation rate, about a 4-fold improvement. And significantly, if we are just putting random peptides on, we don't see that signal. What's even more surprising is APN2 SHL tagging by itself doesn't result in any AT increase, suggesting there's some sort of synergy happening here that is allowing for MSH6 and APN2 to increase the AT rate together. And so this was great, we're super happy. We increased the AT mutation just by 4-fold, but that wasn't so wonderful when we looked at it relative to C and G, because the CG mutation rate was around 10 to the minus 3, and even with our 4-fold increase, that's still about 1,000-fold lower than what we were achieving with the deaminases. So this brought me to part 2 of enhancing mutation diversity. And now I kind of return to the fact that the deaminases are doing all of the work, really, and that's where we've seen the most success. Wouldn't it be nice if there was a adenosine deaminase that could help out on the AT mutations? And it turns out there are adenosine deaminases, but sadly they only work on RNA. And so this TAD-A is from E. coli, and it deaminates adenosine to inosine and tRNA. But about a year and a half ago, David Liu at Harvard this didn't stop them. They just decided to turn TAD-A into a DNA editing enzyme using directed evolution, allowing TAD-A to deaminate adenosine to inosine on DNA and not just RNA. And they engineered a variant called TAD-A star, which they combined with TAD-A in a heterodimer, which they then fused to Cas9 and used as a base editor. So we took that from the Liu lab and thought, wonderful, we'll just fuse it to T7 and this will solve all the problems. But it wasn't so wonderful when we fused it to T7 because without targeting, there was almost no change in the TAA reversion rate. Just a reminder, now we're looking at this is fluctuation data again. So that was kind of disappointing, but that in their paper, they had kind of delineated a strategy for evolution and when they were doing the evolution of Cas9. And so I thought perhaps we could come up with a strategy to evolve TAD-A for targeting by T7 and improve the enzyme for activity in our system. So we took the TAD-A star heterodimer that the Liu lab engineered and fused it to T7. And with the URF3 gene having a stop codon, if a TAD-A star variant can correct that stop codon, it will fix the URF3 gene, allowing growth on uracil. If we construct libraries of these variants using traditional ex vivo mutagenesis, then one can envision repeating the cycle many times, which slowly result in TAD-A variants that are better at correcting stop codons and better at mutating at A and T bases. Also note briefly that we're using traditional directed evolution instead of the in vivo mutagenesis tools here because our in vivo mutagenesis tools weren't completed, so we didn't want to use those yet. So we did this, and what we saw was with the parent, there was almost no mutations in the first round, but by round 4, we had this massive increase in the number of mutations forming. So we sequenced some of those and found a consensus of 4 additional mutations in blue that we pulled out on top of the 14 that the Liu lab had engineered in the TAD-A. And wonderfully, when we expressed this in the yeast with the T7 fusion, we saw around 1,000-fold on and off target ratio now, which was really encouraging. And we put this into the NGS assay just to double check, and we now see an increase in T to C mutation rate. In blue, that blue curve is the T7 targeting with the yeast engineered TAD-A versus in yellow, just T7 alone with the CDA1. And all this carried through also for the A to G mutation rate. You can see while the signal is kind of pretty close to the noise limit of what we're able to detect, there's still an unambiguous increase in the A to G mutation rate following the T7 promoter, which was very encouraging. Finally, we wanted to check, does this yeast TAD-A play well with our SHL strategy in terms of the GA mutation rates that we had improved? And you can see in the absence of targeting, we just have a flat line. The yellow and blue curves are the yeast TAD-A and CDA1 expressed alone, but when we have the SHL in combination with the yeast TAD-A and our CDA1 targeting in the black curve, we do get an increase in the GA signal compared to the others, indicating that SHL strategy can work on top of all of this additional engineering. And so the strains that this resulted in have the following construction. We have an inducible promoter driving our engineered TAD-A enzyme fused to T7. We have another inducible promoter driving the SH3 domains fused to CDA1 fused to T7. And then we have these two SHL targeting systems for the native yeast proteins. And the overall mutation profile that we now achieved was very significantly biased to C, but we've been able to really increase the G, A, and T substantially over what they started out as. And one can envision that if we were to, pursue some of the strategies that I've outlined, that the A, T, and G rate could be further balanced to make this really equal distribution of mutation. So with this yeast type mutation platform, what I'll refer to as YHM later, as kind of analogous to somatic hypermutation, but in yeast, we now went on to our third aim, which was to apply in vivo immunogenesis strategy to evolve proteins. And the protein that we wanted to evolve was a fluorescent protein initially. And this is because fluorescent proteins are very scientifically important. A Nobel Prize was awarded for GFP a while back. And fluorescent proteins are a common model for protein engineering. In addition, fluorescence generally is a commonly used tool in tagging of cells and proteins and enzyme engineering. So fluorescent properties are a common strategy for engineering and for as a proxy for engineering. The first challenge we set our system to was just simply correction of stop codon in mCherry. So we edited an integrated copy of mCherry from an asparagine to a stop codon. And if the in vivo immunogenesis system works, then we expect that we can correct that stop codon simply just by growing cells. The old method of ex vivo mutagenesis would require one to take the gene of interest, mutagenize it outside of the cell, and then transform it back into the cell. And the system that we built, all we need to do is add an inducer, walk away, grow the cells, and come back and sort, because we've engineered all these mechanisms into the cell to carry out the mutagenesis for us. So we carried out this, we created a library using the strategy, and then we sorted for mCherry expressing cells. This is just a representative sort gate for what we were trying to grab from the library. When we analyzed the sort results, we saw a really significant shift in the number of mCherry positive cells. And when we sequenced some of those cells, we indeed saw mutation correction within mCherry, supporting the conclusion that our system is doing the targeting. Although I sorted for like 2 years prior to the final iterations of the system, so we're fairly confident that our yeast hypermutation platform is inducing the mutations and it's not some sort of off-target, non-specific mutation occurring. We also looked at what sorts of mutations were occurring outside of our target region, and we were able to see pretty good mutation diversity at sites other than the stop codon, with a mutation rate of around 1 mutation per kilobasis sequence. In addition, we compared the system to other state-of-the-art Cas9-based targeting systems in the literature, and we are achieving a comparable stop codon reversion relative to a recent paper that used Cas9 to target mutations in E. coli. So with this success, we then turned to trying to evolve a more complex trait. And the trait we were interested in trying to evolve was altering the spectral properties of M-cherry. So for fluorescent protein, there's some excitation spectrum that when you shine a laser, it will make the protein emit at a certain wavelength. And the emission spectrum, shown here in red, follows some characteristic shape. A red-shifted protein we would like to see would have a shifted emission spectrum to the right, indicating that there's been a red shift in the emission profile. So we follow the same strategy that I described before, where we're using our in vivo mutagenesis system to induce mutations in a labor-free manner. And then we sorted cells, this time kind of off a diagonal. So 616 and 660 are just emissions of light. And so M-cherry falls along a very particular diagonal of 616 and 660 emission. In order to get redshifted versions of M-cherry, we need to select cells in this representative box, which have an increase in 660, indicating an increase in the amount of redshift signal. When we carried out these sorts, we sorted for about 5 rounds of enrichment, because each round gives us about 100-fold individual enrichment. And we eventually pulled out an F23S mutation in M-cherry, which you can see is off-shifted on the diagonal. When we analyzed this mutant via spectral imaging, we saw a redshift, a general redshift in the peak, and about a 5 nanometer redshift in the overall emission max. And this really is the first example of an engineered in vivo mutagenesis system being applied to evolve a novel non-growth linked trait. And that kind of brings me to the summary slide where, in the first part of the talk, which I maybe sped through a bit since I'm at only 43 minutes, we engineered these systems for and carefully analyzed mutations that we were developing using our T7 targeting strategy. Following that, we took 2 different strategies to enhance mutation diversity using this modular SHL domain strategy and engineering additional deaminase to enhance the AT mutation diversity. And finally, we showed that these strains can be applied to circumvent the traditional cycle of directed evolution and allow us to just grow cells and introduce mutations in a very specific targeted region without the need to isolate DNA ex vivo and retransform it into cells. And so with that, I have very many people to thank. First, the Smoky Lab has been wonderful. I've appreciated how kind everyone has been, how many friends I've come away with from the lab, even people who are gone and alumni I've been able to stay in touch with throughout the years. The Stanford Bioengineering staff has been wonderful in keeping on top of all of the recent graduates, or soon-to-be graduates, and the Stanford Shared Facts Facility, as well as the PAM facility here, have been instrumental in carrying out some of these next-gen sequencing assays and helping me get trained to sort. Sometimes I wish I hadn't been trained to sort. That's okay. I have so much gratitude to Christina for her support. She supports all of us in whatever we want to do in the lab, whether that's due to internships or pursue completely independent projects that she's not sure will pan out in the end. All of my committee members, Jennifer she taught one of the first classes I took at Stanford, and I've met with her many times to talk about this project. And we have a collaboration that hopefully will come through on evolving antibody fragments. Dan and Daniel and Posu, thank you all for your input. Some of you have been with me since calls, and others more recently have been really valuable and positive influences on the progress of this work. A special dedication to Christina. So I dug this email up from a few years back. Back in 2011, actually, when I was spamming professors to try and find summer research positions, turns out she didn't reply to this email. That's okay, because it works out. I actually had sent a similar email to Jennifer. Identical email with the name swap. You didn't respond either. So,
But you sure you didn't have the wrong name?
Yeah.
You better not get a response.
I was sending a lot of emails. I'm so grateful to the postdoctoral mentors who have included me in some of their projects and really been inspirations to the level of efficiency and ability to just power through whatever challenges come in the way. So, my Smoky Lab collaborators, Usman and Desi, were really instrumental in carrying out some of this evolution. They were incredibly helpful in the fall and during a lot of my internship absence, in keeping things going while I was, coming in at night and trying to keep progress going forward. I have a kind of trouble once I pick something up, I don't want to drop anything, so it ends up juggling, juggler with 4 balls and 5 and 6, and you don't want to drop the balls, which causes problems. Tim, Prashant, James, and Colin all have been wonderful metabolic engineering side collaborators and have helped me get some reagents in the lab as well. I made many friends at Stanford. I did get permission to use that photo. And it's really been a fun time. I just briefly mentioned that we installed a garden a few years back, which we thought would be lovely to grow vegetables in, and then left winter break, and on return, came back to thousands of flies that decided the garden was lovely as well. And so the result of that is the biohazard bags being taken away with all the dirt. Finally, have family acknowledgements. My parents really have been inspirational, and they've shown me how just following and pushing for your passion can take you anywhere. And then, maybe I should have written this down. Brian, I met you in my first year here towards the end. And while I think I would have graduated faster, it reminded me that there's a lot more to life than just working. And so that, I'll leave this just on a picture I drew in my first year here of the data I hope to generate prior to having any data. And I'll be happy to take questions. Yeah.
What's the standard mutation rate for somatic hypermutation?
It's above 10 to the -3 mutations per base pair division. So it's very high, yeah.
But that seems to be about what you were getting as well.
The only caveat on our mutation rates is that they're not per division. Okay. They're about 4-fold lower if you break it down by division.
Okay, I'm curious. So you evolved the A to T mutagenizer. Has anyone tried to evolve the yeast homolog or even AID to go C, G, AT or broaden that spectra?
C to G, there's several problems with the DMAs. They have sequence specificity bias, so probably you can see in some of the plots there are dots that are way high up because there's preference for where the sequence is binding. People have created hyperactive variants, but the problem with those is they then non-specifically increase the background rate as well. Yeah, if that answers your question.
So no, so not really.
Kind of, but they didn't. But they're on. You don't want to just put them in necessarily.
Your plots of background levels you have, is that primarily due to sequencing errors, or is that really the background?
It's exclusively sequencing errors. So via MySeq, the reason I say that is first because in some of these preparations, let's see, we have, maybe this actually isn't helpful. We have basically sequence on NextSeq and MySeq, and sequencing profiles are massively different, which leads me to think that it's sequencing error. And yeah.
Are you trying anything with using little eyes?
Yeah, we never tried UMI, in part because our sequencing depth was already around 300,000, so to use UMI, we'd have to multiply that by 3, which would then be really deep. Too deep. Yeah, Kyle.
In terms of, so for both of the modifiers, you had the SHL fragment. So I would imagine that because each of those fragments sort of biases its own deamination, and you're trying to expand the repertoire of mutations to get sort of, ideally 25 would be perfect, right? Is it possible, could you have used another sort of system, sort of two-component D-kind system?
What kind of system?
Well, basically, that you could guarantee that both of those domains would hit. Right now, it could be
Orthogonal binary domains.
Yes.
I did consider that. I spent about a year and a half on the SHL strategy. We constructed like 60 strains in combination, and what we were gaining was just so little that, and you can see doing the evolution of the TAD-A gave us way bigger improvement, and then we spent about like 4 months on that. Okay, with that, it's been on the cake photo. So if anyone would like to come to, we have a joint reception because Calvin back there defended earlier today, and on the left is, I think, some of my, well, that's definitely my data, and on the right is his work with computational design of RNA switches. So you're all invited to this, assuming I pass. Okay, yeah.
We are, right? We're saying.
We have till 4:45.
Well, is the scale on the y-axis? Well, we always have to be able to see.
You don't want to know. The one log one is not log.
I didn't look at that.